ATP and Phosphagen system summary. What is really important things of the Phosphagen system and what is the importance of the Phosphagen system? Because the Phosphagen system including ATP hydrolysis, I would like to mention ATP hydrolysis shortly. I mentioned that the cell's ATP concentration is fairly well maintained, even in skeletal muscle during the most intense contraction. Many exercise physiology textbook authors have described ATP as the energy currency of the cell. But if you think, oh, currency is kind of money, so I think, like money, we can save the money at the bank. So we can save ATP in our body as much as possible. Oh, that is not really correct. When the body has been given too much energy from overeating, cells do not increase their store of ATP and for a more complex reason would impair the ability of the cell to transfer phosphate group between molecules of metabolism. To maintain the cellular concentration of ATP, there are three energy systems to maintain their cellular concentration of ATP. Three energy systems we usually call ATP-PC system, glycolysis, and cell respiration. However, there are two more reactions like adenylate kinases and AMP deaminases reaction. We had a better name, a Phosphagen system instead of ATP-PC system. And there are two reactions of carbohydrate which are glycogenolysis and glycolysis. So we had a better call glycolytic energy system or glycolytic metabolism instead of glycolysis. And respiration occurs in only mitochondria, so we had a better name mitochondrial respiration instead of cell respiration. You watched the Wingate muscular power test video, right? How was it? It is all out exercise, right? So how our body can maintain the cellular ATP concentration during this kind of all-out exercise. The Phosphagen system has a responsibility. How Phosphagen system maintain the cellular ATP concentration during high intense exercise? Or actually the initiation of all exercise is supported by the Phosphagen energy system. Did you remember our lecture video entitled Creatine Kinase Reaction lecture? Using Hess's law, Dr. Gon showed add two reactions, which are hydrolysis of ATP and hydrolysis of creatine phosphate. Because when the reactions are coupled, the two free energy exchange can be summed, resulting in a net free energy exchange that approximately zero or is at equilibrium. If the concentration of the substrate increase, then there will be a change in free energy favoring the reaction to form ATP. Conversely, if the concentration of the product increase, then there will be a change in the direction of the reaction to favor the formation of creatine phosphate. This is a consistent scenario in metabolism in cell. Reactions that require free energy are coupled to reactions that release free energy. This figure reveals that in many instances, reactions releasing free energy are coupled to the formation of ATP. ATP formation can then be thought of as a process that hence free energy. The free energy released from the breakdown of ATP is then used to drive chemical reactions that require free energy. ATP should therefore be viewed as an important molecule involved in the flow of free energy from reaction that release it to reaction that use it. The creatine phosphate reaction is the most rapid means to regenerate ATP and is catalyzed by creatine kinases. During muscle contraction, when there are transient increase in ADP in the location of a contractile filament, the direction of the creatine kinase reaction favor ATP production. However, the ATP production capacity of the creatine kinase reaction rely on store of creatine phosphate, which approximately 24 millimole kilogram when. One additional enzyme catalyzed reaction exists to regenerate ATP, and the enzyme involved is called adenylate kinases. sometimes myokinesis. The adenylyl kinase reaction is similar to the creatine kinase reaction is that it is near equilibrium. This reaction also serves to produce the activator AMP of the allostatic enzyme phosphorylases for glycogenolysis and phosphofructokinesis for glycolysis, thus stimulating increased carbohydrate metabolism. The importance of the Phosphagen system is that it can regenerate ATP at the highest rate possible. During exercise that demand ATP production in excess of ATP supply from mitochondrial respiration or glycolytic energy system, the creatine kinase reaction enables additional ATP to be produced to meet the ATP demand of muscle contraction. However, the finite intramuscular store of creatine phosphate can be depleted during intense exercise is as little as 10 seconds. The AMP deaminase reaction, although not regenerating ATP, is also an important component of the Phosphagen system. This reaction converts the AMP produced from the adenylyl kinase reaction into IMP, thus accounting for the loss in total muscle adenyl molecule during intense exercise. However, the more important reason for explaining this reaction is that it is result in the production of ammonia. Ammonia can also be produced via the catabolism of amino acid.